Wolbachia is a genus of intracellular bacteria that infects mainly arthropod species, including a high proportion of insects, and also some nematodes. It is one of the most common parasitic microbes and is possibly the most common reproductive parasite in the biosphere. Its interactions with its hosts are often complex, and in some cases have evolved to be mutualistic rather than parasitic. Some host species cannot reproduce or even survive without Wolbachia colonization. One study concluded that more than 16% of neotropical insect species carry bacteria of this genus, and as many as 25 to 70% of all insect species are estimated to be potential hosts. The genus was first identified in 1924 by Marshall Hertog and Simeon Burt Wolbach in the common house mosquito. They described it as a somewhat pleomorphic, rod-like, gram-negative, intracellular organism apparently infects only the ovaries and testes. Hertog formally described the species in 1936, and proposed both the generic and specific names. Wolbachia pipientis. Research on Wolbachia intensified after 1971, when Janus Yen and A. Ralph Barr of UCLA discovered that calyx mosquito eggs were killed by a cytoplasmic incompatibility. When the sperm of Wolbachia-infected males fertilized infection-free eggs. The genus Wolbachia is of considerable interest today due to its ubiquitous distribution, its many different evolutionary interactions, and its potential use as a biocontrol agent. Phylogenetic studies have shown that Wolbachia persica was closely related to species in the genus Francisella and that Wolbachia malophagai was closely related to species in the genus Bartonella, leading to a transfer of these species to these respective genera. Furthermore, unlike Wolbachia, which need a host cell to multiply, F. persica and B. malophagai can be cultured on agar plates. These bacteria can infect many different types of organs, but are most notable for the infections of the testes and ovaries of their hosts. Wolbachia species are ubiquitous in mature eggs, but not mature sperm. Only infected females, therefore, pass the infection on to their offspring. Wolbachia bacteria maximize their spread by significantly altering the reproductive capabilities of their hosts, with 4 different phenotypes. Several host species, such as those within the genus Trichogramma, are so dependent on sexual differentiation of Wolbachia that they are unable to reproduce effectively without the bacteria in their bodies, and some might even be unable to survive uninfected. One study on infected woodlice showed the broods of infected organisms had a higher proportion of females than their uninfected counterparts. Wolbachia, especially Wolbachia-caused cytoplasmic incompatibility, may be important in promoting speciation. Wolbachia strains that distort the sex ratio may alter their host's pattern of sexual selection in nature, and also engender strong selection to prevent their action, leading to some of the fastest examples of natural selection in natural populations. The male killing and feminization effects of Wolbachia infections can also lead to speciation in their hosts. For example, populations of the pill woodlouse, Armadillidum vulgare, which are exposed to the feminizing effects of Wolbachia, have been known to lose their female determining chromosome. In these cases, only the presence of Wolbachia can cause an individual to develop into a female. Cryptic species of ground weta are host to different lineages of Wolbachia, which might explain their speciation without ecological or geographical separation. Wolbachia infection has been linked to viral resistance in Drosophila melanogaster, Drosophila simulans, and mosquito species. Flies, including mosquitoes, infected with the bacteria are more resistant to RNA viruses such as Drosophila C virus, norovirus, flock house virus, cricket paralysis virus, chikungunya virus, and West Nile virus. In the common house mosquito, higher levels of Wolbachia were correlated with more insecticide resistance. In leaf miners of the species Phalanoricta blancadella, Wolbachia bacteria help their hosts produce green islands on yellowing tree leaves. That is, small areas of leaf remaining fresh, allowing the hosts to continue feeding while growing to their adult forms. Larvae treated with tetracycline, which kills Wolbachia, lose this ability and subsequently only 13% emerge successfully as adult moths. Musky D. Thorax uniraptor, a parasitoid wasp, also benefits from hosting Wolbachia bacteria. In the parasitic filarial nematode species responsible for elephantiasis, such as Brugia malayi and Vacheria bancrofti, Wolbachia has become an obligate endosymbiont and provides the host with chemicals necessary for its reproduction and survival. Elimination of the Wolbachia symbionts through antibiotic treatment therefore prevents reproduction of the nematode, and eventually results in its premature death. Some Wolbachia species that infect arthropods are